Hi, welcome to this lecture on environmental adaptation for animals. Now we will continue on the thermal relations of ectotherms and how the evolutionary changes happened in the ectotherms. So why we have some animals that can tolerate some temperatures where the other animals will die. And sometimes the comparison is quite difficult to do because the thermal tolerance among the animals is so highly variable. So we have stenothermic animals that are living only at certain temperatures. We have eurothermic animals that are living at wide range of temperatures. We have animals that are living in cold, animals that are living in quite hot environment. We have Antarctic fish that die if you put it in the refrigerator. You have tropical fish like zebrafish. It will also die in the refrigerator because now it's in the cold stress. Actually, the zebrafish is dying in cold stress so well, so if you put it on ice, that might be the best way to decapitation. So there's not a common temperature that where you could test the Antarctic fish and zebrafish. Or there's not common temperature where you could test the cells of these two fishes. And we'll already have learned that, okay, in the Antarctic fish, they didn't have the myoglobin, they didn't have hemoglobin, and that's why, okay, they are not having a lot of oxygen in their circulation, and that's why they can't tolerate high temperatures. But usually the differences are not so clear. And if it's not very stenothermic, then the responses are quite difficult to predict, okay, why it's not tolerating or is it tolerating. And also there are some practical problems in comparison of different animals. First of all, the animals might not tolerate the same temperatures or conditions. And one of the problems in the conditions is that, okay, but they are not tolerating well the laboratories. So in some cases, the animal maintenance is quite difficult to do for some animals. But also the comparative experiments are laborious. So if you have comparing 5 different species, you need 5 times more experiments. And also the link to evolution can be quite difficult to follow. So if you are collecting animals that are living near your laboratory, you will find that, okay, one animal was tolerating well cold temperatures because it has this kind of mechanism. Another animal was tolerating also, but it had another mechanism. So what's the large picture on this? And if you are having rainbow trout, either because it's in comparative physiology, it works as in the same role as in the rat in the medical physiology, or but also because it's using a lot of aerobic catabolism. Then you have Crucian carp that is tolerating well anoxic period and has a huge amount of glycolytic energy production. So what you have to have some kind of reason why you have selected those animals. So you actually, if you are testing that, okay, how the rainbow trout is adapted in different temperatures, you should perhaps compare all kind of salmon species around the world because now they are close relations. And also the problem is that, okay, if you are making these kind of comparative experiments, you have already tested 10 different species with the same setup. Now you want to have one more. It's quite difficult to tell for the people that, okay, what is the purpose of repeating these same experiments again and again. But let's go back on the ectotherms. So they have this thermoregulation with the behavior. The fish were going in certain temperatures, and the lizards were going in the sunlight or in the shadow to regulate their body temperature. And this behavior is demanding energy, so it has to have some kind of benefits. And the benefits can be difficult to establish, but in some cases it's been shown. In the lizards, they optimize the body temperature to maximize the sprinting speed. So those species that try to maintain their body temperature in 30 degrees of Celsius, they are fastest runners at the 30 degrees of Celsius. Then we have animals that are trying to keep their body temperature at 40 degrees of Celsius. They are the fastest runners at 40. And at the same time, their testicles are developing faster at that temperature. Their digesting is working better. Their hearing is working better. So there are several different factors that are affected or optimized because they are having this temperature. But it should be reminded that, okay, not all the animals are doing this. Not all the mechanisms are doing this. So if you have you take a random ectotherm from the forest and test random reaction of the animal, most probably you will not get this kind of optimal correlation. Okay, if you have animals that are tolerating or optimizing their mechanisms, is because they have differences in the proteins. And from that we can get back on the Antarctic fish again. And the Antarctic fish is having this partial compensation of cold acclimation. So it has faster protein synthesis in the liver to compare the other polar species that are living in a little bit warmer temperatures, and it also have the skeletal muscle that can generate more power. And okay, it's increasing because otherwise all the processes are decreased in these very cold temperatures. So it's compensating it. And how you can compensate it? Of course, you can compensate it by modulating the protein structures. You can either have more proteins to do the job, or then you can have differences in the protein structures, either by having different isoform, so you have similar protein with little bit different characters, or then you can have some mutations, so small differences in the protein structure that are affecting on the functional properties. You have one or two amino acids are changed, for example. And one of the things that can be changed is that, okay, all the proteins works better at a certain temperature. So if it's too hot or if it's too cold, the proteins become denatured. They are not working at all. And one classical example of that is on crystalline. So in mammal, if it's kept at refrigerator temperatures, it would get the eye lens will become cloudy. So the crystalline is denatured. The same thing happens also in tropical fish, but it takes a little bit more time, 2 days. And if the lens is cloudy, you can't see anymore. So it's this kind of cold cataracts. But because the crystalline is modulated in the Antarctic fish, this Antarctic toothfish, that's why it's very clear even at -2 degrees of Celsius. So it has some small differences in the crystalline structure affecting that, okay, it can maintain the crystalline invisible and not denatured. Okay, another example is that when we have enzymes, we have learned that enzymes are making some chemical reactions possible. And usually the substrate affinity is the limiting factor for this enzyme activity. So the enzyme is binding substrate before it can modulate the substrate to something else. And depending on how much substrate you have, then this enzyme is either unoccupied or occupied. And if you are increasing the affinity, it will eager bind this substrate. So even in lower concentrations, the enzymes is working. And we have talked a lot of on lactate dehydrogenase also already, and it's converting pyruvate to lactate in aerobic energy metabolism, but it works in both directions. And one of the things that you should remember that it's also modulating NADH, and the substrate affinity is increased in cold temperatures, so it binds more eager pyruvate to enhance the anaerobic metabolism in cold temperatures. And when you are comparing different animals, you can see that there's a lot of variation in these enzyme affinities. So depending on the temperature, they are all getting more binded, or the affinity is increasing at colder temperatures, but they are completely in different scale in desert iguana and in Antarctic fish. And because it's caused by they are different isotomes, so they have different isoform of this protein. Or there can be also some differences in their enzyme structures. And at the normal living temperatures marked over here in blue is that at their normal living temperatures, the affinity is actually quite much the same. And if they would all use the same enzyme structure, they would all either be too high or too low anaerobic metabolism. So that's why they have different isoforms or different modifications in these enzymes that now they can regulate these in their normal living temperatures. But the lactate dehydrogenase is actually tuned quite fine-tuning also. So over here we have 4 different barracuda species, and even they have different lactate dehydrogenase homologs and little bit differences in the substrate affinity. And it's quite fine-tuned because the body temperature differs only 4 degrees of Celsius or 8 degrees of Celsius, and they adapt with these different isotomes. So again, in the blue there are these their normal living conditions, and at those living conditions the enzyme substrate affinity are quite much the same. And so that's why at their natural temperature zone the affinity is quite equal. And moreover, when we have these different isotomes, they also affect on the reaction speed, but let's not go on those. But these enzymes, we have that, and they are modulating these enzymes a little bit, so they have a little bit higher affinity, etc. And usually these enzymes were working at quite large range of temperatures, but sometimes they are not working. We have acetylcholine esterase that is metabolizing acetylcholine. And acetylcholine is a neurotransmitter, and in the synapse, the acetylcholine is metabolized to acetic acid and choline, so the synapse is not continuously active. And in Antarctic stenothermal fish, it's highly dependent on the temperature. So it stops activity completely in plus 5 degrees of Celsius. Well, so the enzyme affinity is very low, dependent on the temperature, and that might be affecting that. This animal, the neurons are not working, and neuromuscular junctions are not working, etc. So it's a quite drastic collapse of the whole animal. And last but not least, we have homeoviscous adaptation. We have learned in the previous videos that, okay, the membrane fluidity was modulated with different kind of factors. We have the number of the double bonds, and we have different kind of phospholipids. We have cholesterol and packing the lipids in microdomains. They are all affecting. And you still, if we are thinking on the where the animals are normally living, the fluidity of these membranes are quite similar. So those in Antarctic fish, the fluidity is around between 4 and 5, whereas also in the rat or turkey it's between 4 and 5, but now around 37 degrees of Celsius. And of course that's essential because, well, now that, okay, these phospholipids, we have the phase transition temperature. So they can either be this gel or then this liquid crystal. And if it gets in the very non-moving, these phospholipids, then these membrane proteins get non-moving and your neural work stops working. So that's why the homeoviscous adaptation is so essential. Thank you.